Hello, I am Sandra Romain, a PhD student under the supervision of Claire Lemaitre, both of us from the GenScale team of the INRIA Center of Rennes. And in this video, I will present you my work on structural variant genotyping. But first, how do we come to study structural variant? So with the increasing availability and quality of sequencing technologies, a lot of effort has been put on intraspecific genome comparison in order to assess the underlying genomic diversity of species. This diversity can be studied by identifying genomic variants, which are classified into three main categories. The first two categories are SNPs and indels, which were the first to be studied extensively due to their small size. The third category represents the structural variants, that I may address as SVs in this presentation, which are bigger in size and are now better studied with long-read sequencing technologies. So SVs are the most commonly defined as variants of at least 50 base pairs, but they can be as large as a few megabases. And as such, they are the main source of genomic variability in terms of the amount of base pairs altered. They are themselves classified into multiple types, such as deletions, insertions, inversions, translocations, or duplications, for example. So once SVs have been identified and characterized, the next step is to genotype them in individuals and populations, as SVs can impact phenotypic diversity and have a role in diseases or evolution. So what does it mean to genotype a SV? Let's say we have a well-characterized deletion and a sequenced individual. The question here is to determine how many haplotypes of this individual carry the deletion. So the most common way to do this is to map the reads on the reference genome and to see how they align on the region of the deletion. There are specific patterns expected for each allele. Here, both patterns are present, meaning that the individual carries both alleles. It is heterozygous for the deletion. So for SV genotyping based on remapping, we consider two main parameters. The first one is the read size. So here we use long reads. And the second one is the SV representation that we use to map the reads on. So one option is to map the reads only on the reference genome. It is the option I illustrated just before. The problem with this representation is that it causes a reference bias in the genotyping. So a second method to avoid this reference bias is to represent both reference and alternative alleles as independent sequences. This is the method proposed in SVJedi, which is a tool that was developed in our team. So as I said, it gets rid of the reference bias, but it shows limitations when we are dealing with close structural variants. As here, we have to choose the haplotype to consider for the nearby SV when we represent the alternative allele. A way to overcome both of these limitations is to use a variation graph to represent the structural variants. So methods to genotype SVs using such a variation graph already exist for short-read genotyping, but to our knowledge, none exist for long-read genotyping. So here, our contribution is the development of the first structural variant genotyper with long reads that uses a variation graph to represent the SVs. And this method we call SVJedi-graph. So here, a quick overview of SVJedi-graph method. So as input, it takes a reference genome SV set in VCF format and long reads. So the first step is to construct the variation graph using the reference genome and the SV set. Then the second step is to map the long reads on this variation graph. The third step is to filter the alignments produced by the mapping, and then to predict the genotypes and output them in a VCF file. There are two key steps in this method. The first one is the variation graph construction. So first, we list and sort all breakpoints for each chromosome. Then the reference sequence of the chromosome is split at each breakpoint position, and the resulting sequence fragments become the reference nodes in the graph. Then between each reference node pair, we add the reference edges. They represent the reference alleles of the SVs in the graph, and as a whole, they form the reference path of the genome. Finally, the last step in the construction is to go over the input variants. For each of them, we add the adequate alternative edge depending on the SV type, as well as an alternative node for the insertions that contains the inserted sequence to represent. So then we map the long reads on the graph using an external tool, which is Minigraph. And we come to the second key step in the method, which is the alignment processing and filtering. So we filter the alignments to only keep the alignments that are informative on the genotypes of the variants. For that, we use two filters. The first one is to only keep alignments that have at least two nodes in their alignment path, as we use the information of covered edges to estimate the genotypes of the variants. And then with the left alignment, we apply another filter in order to guarantee a certain confidence on the edges that are covered by an alignment. We require that the reads align over at least a particular distance before and after the covered edge. This particular distance is controlled by the d/over parameter. And for each edge for each alignment that respects this filter on the edge, we increase the support by 1 for the SV alleles that is represented by the edge. So these steps, after this step, we have for each SV the support counts for its two alleles. And then the last step is to predict the genotypes from the support counts for the alleles by computing the genotype likelihood. So here we consider biallelic SVs and diploid genomes. So we have 3 possible genotypes to evaluate, and we output the most likely genotype in VCF for each variant in the graph. In order to evaluate the genotyping performances of the methods, we use two statistic parameters. The first one is the genotyping rate. So the genotyping rate is the percentage of genotyped SVs over all input SVs. We use this parameter because in our method, we require that SV has a minimum support count of 3 supporting alignments in order to predict its genotype. And then the second parameter we use is the genotyping accuracy. which is the percentage of correctly predicted genotypes over all predicted genotypes. So first, we wanted to evaluate the impact of SV closeness on the genotyping quality. So for that, we simulated 12 datasets on human chromosome 1. These datasets had real deletions, to which we added simulated deletions that were close or overlapping one of the real deletions by a particular distance range. So the distance ranges are showed on the bottom, on the x-axis of the figure here. So on the left of the figure, we have the 6 datasets that contain overlapping deletions, the most overlapping on the left and the less overlapping on the right. And then on the right of the figure, we have the 6 datasets that contain closed deletions. The closest deletions on the left and the less close deletions on the right. Here the results show that for most of the long-read genotyping methods, the more the deletions get close or overlap each other, the more the genotyping accuracy drops, which is not the case of SVJedi-graph. Here we show that SVJedi-graph keeps a stable accuracy on all of the simulated datasets, as well as a stable genotyping rate. Then we wanted to evaluate our method on real data. So for that, we chose the highly curated Tier 1 datasets of the Genome in a Bottle Consortium. So we genotyped these datasets using PacBio CLR reads with our method SVJedi-graph and with 4 other state-of-the-art long reads genotyping tools. Here we showed that SVJedi-graph was able to genotype almost all of the 12,000 SVs with a higher accuracy than the other tools, and in less than 30 minutes, which is at least 6 times faster than the other tools. And even without considering the mapping time for the other tools, our total computing time was still comparable to it or lower, at the exception of SVJedi. And lastly, we wanted to challenge our method on a more complex dataset. So we used the one from the Human Genome Structural Variant Consortium, HGSVC. So on the contrary to the GIAB dataset, which contained only distant SVs, the one more than half of the SVs in the HGSVC dataset were close to another one, and almost a quarter of the deletions were overlapping another variant. So for this comparison, we used PacBio Hi-Fi reads to genotype both sets, and we show that overall, our method performs well on various SV set compositions compared to the other tools, and it does not seem to be impacted by the different genotype distribution, as well as it is able to scale up to the 100,000 of SVs in the HGSVC set. To conclude, I presented here the first variation graph-based SV genotyper that uses long reads to genotype the variants. Our method is fast and more accurate than the state-of-the-art long-read genotypers. We showed that, on the contrary to the state-of-the-art tools, our method was not impacted by close and overlapping calls thanks to the use of the variation graph representation. If you're interested, we also present additional evaluations of our methods on ONT reads and on its robustness to noisy data in our paper. So SVJedi-graph is available on GitHub and as a bioconda package. And lastly, I would like to thank GenWest, the INR, and the SFBI. And here are the references of this presentation. Thank you.